How does a single inherited change continue to shape health? Across multiple generations, gradually altering brain function long before symptoms appear, Huntington's disease offers a clear example of how a specific genetic expansion influences neurological pathways over time. The condition arises from variation in the HTT gene, where repeated DNA sequences increase beyond a functional threshold. This expansion affects cellular behavior in the brain, particularly within regions that coordinate movement and cognition. Examining the disorder across families and populations reveals how its prevalence reflects ancestry, migration patterns, and historical gene flow. Through molecular mechanisms, geographic distributions, and the long arc of inheritance, Huntington's disease demonstrates how one genetic alteration can progress silently through generations before, emerging as a clinical condition. The concept of Huntington's disease begins with variation in a single gene known as HTT, located on chromosome 4. This gene contains a sequence of repeated DNA units, identified as key G repeats, which encode the amino acid glutamine. In most individuals, the number of repeats falls within a stable range that allows normal protein function. However, when this repeat count rises beyond a critical threshold, the resulting protein develops characteristics that influence cellular behavior in specific regions of the brain. The primary mechanism involves an expanded polyglutamine segment that alters the protein's stability and its interactions within neuronal environments. This change contributes to cellular stress responses, disruptions in gene activity, and impaired communication between neural circuits. Evidence shows that these effects are especially concentrated in the basal ganglia, a region responsible for coordinating movement and certain cognitive functions. As neural cells within these areas become increasingly vulnerable, gradual degeneration follows, leading to the characteristic features of the condition. Distribution patterns of CAG repeat lengths vary across global populations. Some lineages exhibit higher baseline repeat numbers, while others show greater stability across generations. These differences reflect historical gene flow, founder effects, and the demographic expansions that shaped regional ancestries. In contrast, populations with lower repeat counts display reduced prevalence, indicating how genetic variation aligns with migration histories. Now, intergenerational transmission plays a central role in the expansion process. Repeat numbers may increase when passed from parent to child, particularly through paternal lineages. This phenomenon influences both the age of onset and the long-term distribution of the disorder across families. Today, modern genetic analyses provide clearer evidence of how these expansions propagate through time, allowing researchers to trace the condition's movement across generations. This chapter, therefore, outlines the molecular basis and population-level context through which the HTT gene shapes the emergence and persistence of Huntington's disease. The neurological consequences of Huntington's disease arise from gradual cellular loss within brain regions responsible for movement, coordination, and cognitive regulation. The basal ganglia, particularly the caudate nucleus and putamen, show the earliest and most consistent vulnerability. These structures depend on balanced signaling between excitatory and inhibitory pathways, and disruptions in this balance contribute to the characteristic motor changes associated with the condition. The underlying mechanism reflects cumulative stress placed on neurons exposed to expanded HTT proteins. These proteins interfere with normal gene activity, protein turnover, and intracellular transport, creating an environment in which cells become progressively less resilient. Evidence from neuroimaging studies illustrates early reductions in regional volume, even before outward signs appear, indicating that the degenerative process begins long before clinical recognition. In contrast, cortical regions display a more gradual involvement, aligning with later cognitive and behavioral features. Distribution patterns of symptom onset vary across individuals and populations. Differences in CAG repeat length strongly correlate with the age at which neurological changes begin. but additional modifiers influence the rate of progression. These factors include regional ancestry, background genetic variation, and environmental elements that shape overall cellular health. Today, longitudinal studies allow researchers to map these changes with increasing precision, identifying how specific neural circuits respond over time. The progression follows a consistent pattern. Motor coordination becomes increasingly impaired as basal ganglia degeneration intensifies. Cognitive processes linked to planning, memory, and decision-making gradually shift as cortical pathways become affected. By comparison, emotional or behavioral changes emerge as downstream consequences of these structural alterations rather than independent processes. This chapter therefore outlines how the neurological landscape of Huntington's disease reflects a sequence of cellular disruptions, regional vulnerabilities, and population-level variation, offering a structured foundation for understanding its broader distribution and ancestry-related patterns. The global distribution of Huntington's disease reflects differences in ancestry, historical gene flow, and the demographic pathways that shaped modern populations. Prevalence is highest among individuals of European descent, where specific lineages carry HTT alleles with longer baseline CAG repeats. These expanded repeat ranges increase the likelihood of further intergenerational growth, influencing the number of affected families across regions. In contrast, many East Asian and Sub-Saharan African populations exhibit shorter, more stable repeat lengths, resulting in lower overall prevalence. Evidence from population genetics supports a founder effect model in several areas. Certain European communities possess ancestral haplotypes that predispose the HTT sequence to expansion over generations. These lineages can be traced through documented migration events, including movements during the medieval and early modern periods. By comparison, populations with more genetically stable HTT loci show less evidence of rapid repeat expansion, suggesting differences in background genomic architecture or long-term demographic histories. Now, global distribution also reflects broader patterns of migration. The spread of European ancestry through colonial expansion introduced susceptible haplotypes into the Americas, Australia, and parts of southern Africa. Today, regions with higher proportions of European-derived lineages exhibit prevalence rates consistent with their ancestral origins. However, detailed analyses reveal additional variation shaped by local demographic processes, including population bottlenecks and admixture. Ancient DNA studies provide complementary evidence, although direct identification of Huntington's causing expansions in ancient samples is limited by preservation constraints. Haplotypes associated with CAG instability can be mapped through historical gene flow. These patterns illustrate how long-standing demographic structures influence modern prevalence. The distribution of Huntington's disease therefore reflects interactions between genetic predisposition, ancestral lineages, and historical population movements. This combination of molecular variation and demographic history explains why the condition remains concentrated in some regions while remaining rare in others, offering a broader context for understanding its presence in human populations. The evolutionary context of Huntington's disease centers on how expanded CAG repeats in the HTT gene persist within human populations despite their severe clinical consequences. The condition typically appears in mid-adulthood, after reproductive years for many individuals. As a result, natural selection exerts limited pressure against the allele, allowing its transmission across generations. This timing creates an evolutionary environment in which deleterious expansions can accumulate without strong selective removal. The mechanism of repeat expansion plays a central role. KG sequences are inherently unstable during DNA replication, particularly within paternal lineages, where sperm production involves continuous cell divisions. This instability increases the likelihood of further expansion during transmission, gradually shifting repeat lengths upward across generations. Evidence shows a strong correlation between initial repeat size and future instability, creating a dynamic in which certain lineages develop progressively higher counts. By comparison, populations with shorter, more stable repeats display reduced rates of expansion. This stability may reflect long-term demographic histories or genomic contexts that limit repeat growth. Now, these differences influence prevalence patterns, connecting the evolutionary behavior of the gene to regional ancestries and historical population structures. Some hypotheses suggest that slightly expanded alleles, still below disease thresholds, may influence biological processes in ways that were selectively neutral or mildly advantageous in past environments. However, current evidence for such benefits remains limited, and the primary explanation for persistence remains the delayed onset that minimizes selective pressure. Today, evolutionary genetic models help clarify how repeat expansions behave within different demographic settings. Factors such as population size, mutation rate, and historical gene flow shape the long-term distribution of HTT alleles. These patterns illustrate how evolutionary mechanisms, rather than recent environmental forces, govern the persistence of the mutation. In this context, Huntington's disease reflects a broader principle. Variations that emerge late in life can endure through lineages, shaped by inheritance dynamics rather than direct selection pressures. Modern research on Huntington's disease combines molecular, clinical, and population-level data to clarify how the condition progresses and how future interventions may alter its course. Advances in genetic screening now allow precise identification of CAG repeat lengths, enabling clear assessments of risk across families. These tools also reveal subtle intergenerational changes, contributing to broader population-level analyses that track the disorder's distribution and long-term behavior. At the cellular level, studies increasingly focus on how expanded HTT proteins disrupt fundamental processes such as gene activity, protein folding, and mitochondrial function. These disruptions generate stress responses that accumulate over time, particularly in vulnerable neuronal populations. Today, imaging technologies and biomarker studies help map these changes before symptoms emerge, offering new ways to evaluate disease progression and measure the effectiveness of potential therapies. Therapeutic research includes approaches aimed at reducing production of the mutant HTT protein, stabilizing cellular environments, or improving neuronal resilience. Techniques such as gene silencing strategies and RNA-based treatments illustrate the direction of current investigation. In contrast, supportive treatments focus on managing symptoms by addressing movement, cognition, and day-to-day functioning. While these approaches do not halt the underlying process, they shape quality of life, outcomes, and help track patterns of progression across diverse populations. Population genetics continues to clarify how different ancestries influence prevalence, highlighting the importance of historical gene flow and demographic structures. These insights allow researchers to understand why prevalence shifts across regions and how long-term lineage patterns continue to shape modern distributions. In summary, current research reflects a combination of molecular advances, improved screening, and detailed population-level studies. Together, these approaches offer a clearer picture of how the disorder develops and how future interventions may alter its trajectory, providing a broad scientific context for understanding Huntington's disease.